Brilliant. So for anyone else who's just joined us, hi, my name's Evie. I'm a research assistant at the Museum of Zoology. So I've been working on the Butterflies Through Time project, of which we are starting our Researchers' Stories series. So this is our third talk in the series, where each week we listen to a different butterfly researcher talk a little bit about their research and what it means for the wider butterfly conservation. So this series will be running every Wednesday at 7 p.m. until the 8th of June. So every Wednesday, on 6 consecutive Wednesdays until the 8th of June, on the second week of June. So this is our, oh, have I learned something? Oh, no worries. This is our third talk, so we're halfway through the series. And we're just about to hear from Galina Jonsson in a moment. So Galina is a PhD student at Imperial College London and has been working closely with butterflies collections at the Natural History Museum in London. In a moment, she's going to be discussing the Natural History Museum's work to digitise its butterfly collection and how examining previous periods of climate change and habitat loss can help us better understand insect declines today. So I think that's all from me. Galina, over to you.
Thank you. Let me just share my screen, hopefully. Does that look right?
Yeah, all visible.
Awesome. All right. Thank you very much, Evie, and thanks to the whole Butterflies Through Time team for inviting me. So I'm Galina. I'm a PhD student based at the Natural History Museum in London. I'm also affiliated with Imperial and UKCEH. And today I will be talking about the unique and frankly extraordinary resource natural history collections can be to further our understanding of biodiversity today, as well as to curb current biodiversity loss worldwide. So I will be covering 3 broad topics or sections, if you like. Firstly, why we should even care about collections. And this is from a scientific perspective rather than a purely aesthetical or historical. Second, I will be talking about the Natural History Museum in London's journey to turn its 80 million or so specimens that's held at Natural History Museum into digital format. I'll also touch upon what we've learned from that process more generally. And then finally, I'll go over a bit of my PhD research where I combine contemporary butterfly records with data from collection specimens to extend the population time series backwards in time, that is. So let's start with the first section. Why we should even care about collections, or rather why we should care about historical states of wildlife at all. So as mentioned by Jamie last week, shifting baselines is where ecological expectations decrease with each generation, because the baseline biodiversity state becomes that experience within each generation. Or to put it another way, as actual biodiversity levels have decreased over time, that we do know, we get a kind of generational amnesia, which results in inappropriately low baselines because we only base the baselines on what we have experienced within our own generation. And I often see the windshield phenomenon being used as a kind of proof for shifting baselines. And that is the claim that fewer insects accumulate on windshields today compared to some decades ago. But I was barely born back then and certainly did not drive. So I would like some more robust evidence, wouldn't you? So here are a few news headlines exemplifying that even in countries with some of the best biodiversity data, like the UK is, the baseline against which we measure change is the 1970s, or in many cases actually later. So in other words, we only really know about the parts of the graph indicated in green here. And this graph shows global average temperature anomaly, difficult word, from the year 1850. And yeah, okay, so the part of the time series for which we have biodiversity data, the green part, fair enough. It captures the start of the kind of clear directional warming we started seeing in the 70s, 80s. But we ignored this whole part of the time series when, at least in the UK, most large-scale landscape transformations happened. So that includes, industrialization, deforestation, the huge agricultural intensification that we saw, especially around the 50s, increased pesticide use, etc. And I reckon that these factors must have affected wildlife populations, but without species trends prior to the 70s, we simply can't say for sure. And the reason for why we're still largely limited to this 1970s biodiversity baseline is that this is when most species monitoring schemes began popping up. So for butterflies, we're talking about the UK Butterfly Monitoring Scheme, UKBMS for short, which began in 1976. And that's basically where fabulously committed individuals go to the same place multiple years in a row and record butterflies in the same way. This produces so-called standardized data with a known kind of recording intensity and methodology, which is less statistically tricky to work with. So kind of to summarize, the 70s is our biodiversity baseline because of historical data deficiency. Before moving on, I just quickly want to mention that we also have many more recent and amazing citizen science data sets, including iRecord, where anyone can report a butterfly sighting using a smartphone application. Right, and then just like to put it into some perspective, this 1970s biodiversity baseline that we currently have. The previous graph I've showed you showed global temperature anomaly over the past 200 years. This graph shows the same thing over the past 12,000 years, if my math is correct. So through using tree rings and ice cores, climate scientists have been able to extend these kind of climate time series tens of thousands of years back in time. And this in turn as well has been able to prove the human action impact on climate change or on climate. So the biodiversity equivalent then of tree rings and these ice cores is of course natural history collections. We do have tons of specimens that date prior to the 1970s, but still we don't really use them very much. Why, you might ask? Because we must first turn physical specimens into data that we can analyze, and that we do through a process that's called digitization. Sorry, just grabbing some water. Now we're moving on to the second part of this talk, digitizing the Natural History Museum in London. So back in 2013, the museum started a pilot project called iCollections with the aim to develop quick and efficient ways to digitize large collections. Natural History Museum has around 80 million specimens, of which around 30 million are pinned insects. The iCollection kind of pilot mass digitization project chose to focus on the approximately 500,000 specimens that are British butterflies and larger moths, as well as Irish actually. And as you might imagine, or as you can see here on the slide, it was a large and quite pricey project that spanned 3 years and involved up to 8 full-time staff to do the digitization, that is. So why would they start with the UK butterfly specimens when, you know, in the whole of the museum, there's around 80 million specimens in total? Firstly, because people love butterflies and have done for a long time. So compared to most other insect taxa and, you know, taxa across the board, they're very well represented in natural history collections. So by digitizing them, we would get a good data set to make inferences and learn from. Secondly, there's only around, depending on how you count, around 55 resident butterfly species in the UK. And again, compared to other insect groups, this is a very manageable number of species. Then another reason is that they're also really good indicators of wider ecosystem change, because they have 4 different life stages, as you can see here on the slide. Unfortunately, the adult butterfly is not a native. Butterflies also, well, resident butterflies in the UK go through all 4 life stages at least once within a singular year. So, oh, and one more thing, that the different life stages can also serve as food for various different, like, higher up organisms in the food web. I just have loads of reasons for why they chose butterflies. Another reason is that these 4 different life stages have different needs in terms of foods and climatic conditions. And I hope this is the last point I have. So yeah, finally, the 55 or so different resident species also differ in those needs, the foods and the climatic conditions. So for instance, some of the species have really picky eaters when it comes to caterpillars that would only eat one specific host plant. Others might be more generalist, as it's called, and can eat a range of different host plants and survive in a range of different climatic conditions. But regardless of which species it is, if you remove the food or climatic conditions become suboptimal for a specific species life stage, it won't survive. Easy as that. And then can't continue the rest of the life cycle. And this can have wider ecosystem effects by, for instance, not providing food for birds. Is that a blue tit? I'm not very good at birds. But returning back to the year 2013, iCollections, the project began, and it involved 5 main steps. First, carefully remove the label from the pin with the specimen on the pin. Second, place the specimen and labels neatly on a tray. Take some really high definition photographs of everything. Fourth, this is the step that takes the most time. So extract all of the metadata from the labels. So the labels will say what, when, like what species is it, when was it collected, where was it collected, by whom, etc. You know, some specimens have small essays attached to them about the circumstances, etc., that they were collected in, which is lovely. But it takes a long time to kind of like transcribe all of this by hand. And then the stage that often takes the longest is to georeference collection localities. So as we're working with historical specimens, oftentimes they were, not often, but sometimes, quite often, they were collected in towns that might no longer be called the same thing, might emerge into another town. So you also have to kind of translate whatever it says on the label in terms of where it was located and do some detective work. and translate that into GPS coordinates that we can put on a modern digital map. The last step, the rewarding step, is publish the specimen record, make it freely available on the NHM's data portal. Here I have an example of some of the information that's attached to a singular specimen of a peacock, which apparently was collected in Bexley. There's a lot more information for each specimen. And you can go on the data portal, and if you want to look at all of the published specimen records so far, they're there. Right, so as mentioned, iCollections was a big and fairly pricey project. So 3 years, up to 8 people. It took between 2 and 3 minutes per specimen, and then about £1 per specimen. At the end of the project in 2016, they had digitized around 450,000 specimens. Quite good. But if we compare that to what's on the data portal today we have over 5 million specimens that have been digitized across the whole collection. So we're not just talking butterflies and moths here. But anyway, that is more than 10 times as many during the 6 years after iCollections than were digitized during the 3 years of the iCollection project. So we can definitely conclude that speed and efficiency of the digitization process had definitely increased. And I actually just went on so I put these numbers on the slide here yesterday, and I just logged on to data portal earlier today and saw that this number is no longer accurate, where it's around 5,143,000. Oh, this is a lot of numbers. 900 and something specimens. So just over less than 24 hours, they've added another 400 or so specimens. And yeah, just to kind of highlight that, the museum's collection is incredibly varied, from nanofossils that are barely visible to the human eye to some of the largest animals on Earth, dinosaurs, and the largest animal ever on Earth, the blue whale. And obviously the digitization team have therefore developed different ways of working with each type. But returning to the pinned insects, so one of the many innovations that has made the digitization more efficient is things like this, the ALICE method, which stands for Angled Label Image Capture and Extraction Equipment. Basically, they no longer have to remove the labels from the specimen. You just have all of these cameras around a singular specimen that captures all the information. And although it no longer costs £1 to digitize a singular specimen, the process of digitizing over 5 million specimens has still, cost a pretty penny. So is it worth it? Yes. So according to this report from last year, digitizing the specimens in the Natural History Museum at London collection can benefit a wide range of sectors, from mineral exploration to medicine and agriculture. And importantly, the benefits outweigh the costs by up to 2 billion, that's a B, pounds. So what have we learned from the iCollections project and the associated data set? I'll give you 2 examples here. So Up until 2016, most studies looking at how climate affect timings of egg laying, and other life events typically only covered a few decades. So as we know now, a lack of historical data makes it difficult to kind of put findings into perspective, because the long-term effects of climate change can be obscured by short-term natural variability. So in 2016, Steve Brooks and other museum scientists compared the iCollection butterfly records to historical temperature records and found that before the year 1970 and also before the start of directional warming, nearly all British butterfly species emerged earlier with higher spring temperatures, and some, such as the orange tip and green hair streak pictured here on the slide, emerged as early as 9 days. For every 1 degree increase in temperature, they could emerge 9 days earlier, and that's in spring temperature. So in other words, this paper provided a kind of baseline of normal butterfly response to climate variation, and then we can use this and measure. We can use this and then compare it to changes we see today under rapid climate warming. Jumping forward to just a bit more than a month ago, Rebecca Wilson and colleagues at the museum published this fantastic new resource. So what they did was they used computer learning and the photographs that were taken during the iCollections pilot project and developed an automated, computerized way to detect the specimen in the photo, measure different wing features. It can even identify the sex of the specimens. So now researchers barely don't even have to look at the photos of the specimens to get certain data. And I do believe that this resource will definitely come in handy as more and more collections around the world start imaging their specimens. Right, and now we're already on to the last bit of my talk, where I'll tell you what I've done with digitized iCollections butterflies, and a bit more broadly how they form the basis of my PhD project. But let's start with summarizing some bits from the first section. So we learned that at least compared to other insects, we have a lot of butterfly specimens, as well as contemporary observational record with frankly amazing spatiotemporal coverage. We of course have the iCollections data set, which I just went over, the project where NHM digitized around 500,000 British Lepidoptera specimens, Lepidoptera being butterflies and moths, but we're only talking about the butterflies here. I've also included 3 other collections on the slide here, and if you can find a link for those 3 other institutions where I've used as many specimens as they had digitized when I compiled my data, I'll give you well done if you find a link between those 3 institutions, and I'll tell you what the link is towards the end of this talk. Anyway, in addition to the specimen data, we also have tens of millions of contemporary observational records. So we have the first standardized monitoring scheme, the UK Butterfly Monitoring Scheme, UK BMS for short, which started in 1976. So for butterflies, our current baseline is 1976. Additionally, we have loads of other modern initiatives which produce everything from super standardized data, where standardized is the kind of data where we know exactly how it was collected, why it was collected. But some of these modern initiatives also have very opportunistic data. So data where there was no clear kind of methodology of how to record. So some people might be more interested in colorful species and only go for colorful species. Other people might be interested in getting every single variety of one species. And this is kind of variation we see within natural history collections, as well as some citizen science data sets. And I'll show you an example of the latter on the next slide. Right, so one of my first tasks when I started my PhD was to find a way to integrate, which kind of means statistically combine appropriately, all of these different kind of data sources of butterfly records and use them within one model. But a problem with using data from different sources is that they have different kind of biases. So we wouldn't expect Victorian naturalists to collect exactly in the same way as modern iRecord users, where iRecord is this smartphone applications. And we also expect that neither of those two groups would have the same recording behavior as the UK BMS recorders, who record everything they see in a standardized way. So to be able to account for these differences, I first had to see what the differences were. So I compared individual people or collectors' sampling biases, and I will show you individuals that collected specimens for the Natural History Museum, individuals that do the standardized UK BMS survey transects, and individuals who just use this smartphone application at their leisure and can record, you know, whatever they please without any kind of sampling method. However, for the app, I do believe that you have to include a photo of the specific of your sighting as well, so that the species ID can be verified. Right, so this graph shows that the museum collectors, NHM here, black dashed line, tend to collect only one or very few species at a single occasion, whilst the UK BMS recorders, purple solid line, record multiple species, which makes sense because they record every species observed, right? And then iRecord users, the pink dashed line, fall somewhere in between and vary quite a bit. Again, not too surprising as there's no specific protocol for how to record. Here we can see that both UK BMS recorders and iRecord users tend to record common species, whilst Natural History Museum collectors focus on rare species. And there's also quite a bit more variation among the Natural History Museum collectors. And those were only 2 of 12 different like spatial, temporal, taxonomical biases that I analyzed, and then used my findings to formulate a statistical model that explicitly accounted for these biases between and among data sources. And if you recall that I showed you how climate scientists have used ice cores to extend the climate time series back in time. So they actually had to use quite fancy statistics to calibrate the data from the ice cores to the observational data from, you know, today's weather stations. You can think of it as, ice cores and a weather station. They measure the same thing, but in different ways. So you have to calibrate them statistically. You'll have to trust me on this one, but there were some clever statistics involved to calibrate the differences between these different butterfly data sets that I used to then produce long-term trends across Great Britain. And I did this for each of 53 butterfly species. And I should also say before I show you the results, is that these are what's called occupancy models. So they give you a measure of occupancy. Occupancy is basically the range of a species, and in this case, throughout Great Britain, where if the occupancy is 1, the species is found all across Great Britain. If the occupancy is 0, it's at least locally extinct in Great Britain. If it's 5, it's found on 50% of Great Britain. I think this will make more sense when I show you the some results. This plot, though, shows the annual occupancy change since the year 1900, as averaged across the 53 species that I looked at, again, across all of Great Britain. The vertical dashed line indicates 1976, when UK BMS started, our current baseline for butterflies. And just by looking at it, it doesn't look too bad, in my opinion. The numbers agree too. The average butterfly occupancy in Great Britain decreased by around, I think, 15% or so since the year 1900, when we look at the average across these 53 species. And also, we don't see any overwhelming evidence suggesting that we are suffering from shifting baseline syndrome, because then we would expect to see more of the change happening prior to 1976 rather than after. However, we get a very different impression if we divide the species into whether they are habitat generalists or habitat specialists. You can think of the habitat specialists as perhaps the species that had very picky eaters for caterpillars that only ate one specific host plant species, and the generalists being, the more easygoing ones that could eat a number of different plants, as a generalization. So in fact, we get something that resembles a giant squid, in my opinion. You might disagree. I'm not an expert on giant squids, but the main point here is that the generalists, the blue kind of top dashed line, and these are also known as wider countryside species, they've done great, nearly doubled since the year 1900, whilst the specialist, green dashed bottom line, have not done well at all, and approximately halved. Again, we talk about groups of species here, not individual species. So when one separates the specialist from the generalist, as we've done here, we do see big differences between the two groups. And it's also evident that most of the change happened before our current baseline of 1976, especially between the 50s and the 70s. And I think this is quite interesting because it suggests that perhaps agricultural intensification of the 1950s could be a cause, or at the very least, a kind of tipping point for the subsequent changes. I should also add that these are preliminary results, and I'm currently rerunning and tinkering with the models a little bit. So stay tuned for a paper on the final results. Nonetheless, I do wonder, is it fair to divide species into either habitat generalists or specialists, and then assume that within each of these two groups, all species would show exactly the same long-term trend. It appears that it isn't. So here's two examples that show opposite trends compared to their respective groups. And yeah, perhaps it's clear what occupancy is on these graphs here. So occupancy is measured on a scale from 0 to 1. You can see that the y-axis for occupancy here are different for the two species, but if the occupancy is 1, the species is present on all of Great Britain. If the occupancy is 0, or if it's 0, it's locally extinct, or perhaps even extinct. But if it's low, it's just present in a small proportion of Great Britain. In this case, you can see that the silver wash fritillary starts around 0.2 occupancy. So we would see the species on 20%-ish of Great Britain. Anyway, the point of this slide is that the silver wash fritillary on the left here is a habitat specialist that has increased in recent decades, which is not in line with, you know, the overall group trend where specialists had decreased recently. And similarly, the wool butterfly on the right here, a habitat generalist, has shown quite worrying declines. It's also been suggested that its decline might be due to climate change, which my results here support, because the start of the decline kind of coincides with that of directional warming. But as you can tell, you know, there's a lot to unpack in these data, which is why I've been able to base a PhD thesis, and I'm sure there can be many PhD theses on, you know, this iCollections data set only. And that leads to my conclusions. Firstly, natural history collection specimens, once digitized, can produce long-term trends for as well as kind of timings of life events. Long-term trends for both the timings of life events as well as this occupancy change that I was talking about and showed you in the previous slide. Second, we seem to need these long-term trends because evidently the 70s is a poor biodiversity baseline. We are pretty confident that most change happened before the 70s. Third, as demonstrated by the wool butterfly and silver-washed fritillary, there's a lot of information to uncover in this data set of digitized museum specimens. And finally, it's not quite a conclusion, but this is the answer to the other collections I had on my first slide for my PhD research section. This heat map shows where the Natural History Museum iCollection specimen records were collected. So the collection localities for the digitized specimens from the London Museum. And as you can see, there's a strong bias towards Southeast England. And this can be problematic because in order to make robust inferences and make conclusions, we need specimen records from across Great Britain and ideally across the globe. So yes, iCollections was a fantastic pilot project, and digitizing all of the Natural History Museum in London's specimens can, you know, lead to net benefits in terms of cost of what was it, over 2 billion. But we still need to mobilize resources to digitize all collections, and and that will let us get a comprehensive picture of the past, which in turn will help us protect biodiversity today and moving forwards. And I just want to say, I have by no means done any of this by myself. Everything has been done with my brilliant supervisors and the help of many more. Yeah, thank you for your attention. I'm happy to take any questions. Thank you. Should I finish sharing my screen here?
Yeah, you can end if you want, or we can leave that up if you want people to sort of take note on your screen. That's absolutely fine. Thank you so much, Galina. That was a brilliant talk. I absolutely loved that. I loved the whole journey and how you really took us through everything. Are you all right to? Did you want me to? There we go. If I
there.
Brilliant. Fixed it in the end. Yes, I absolutely love the process that you took us through with the iCollections and your journey, and how exactly you guys got to where you are now. I think that was brilliant. Really did enjoy the giant squid as well. That was a great visualization, along with the Swiss Army knife. It's great collection.
Thank you.
Brilliant. Okay, so while you guys get your thinking caps on for Galina with all your questions, I'm sure you guys have many to ask. I'm just going to post some relevant links to what we've been talking about today, as well as some links regarding the Butterflies Through Time project, and some links that Galina has got for us as well. So they're just going into the chat now. And then I've also got, as I mentioned before, a questionnaire for you guys to fill out if you wish. As I said, it can help us receive feedback so we can improve our events in the future. So that's also just going in the chat for you there. Brilliant. Otherwise, if you could all post any questions you might have in the Q&A, that would be fabulous. Brilliant. I think we had a question early on in the talk. It was about the UK BMS. I believe we've now got a link posted in the chat. I can repost that for anyone who might have missed that. Gina, do you just want to talk about the UK BMS and what that is, and how, if you were interested, you might join?
So basically, you get, it's a big commitment, and a number of people who do this, it's incredible, and I'm so grateful I really thank them for what they do. But you get assigned a transect, or you choose a transect if no one around has like taken it or something, at a specific location, and then you have to go, okay, I don't know this, I'm going to give you some approximate information, but I think you have to go, you walk a kilometer or two, you're meant to record every butterfly you see 2 meters on either side of you. So if you see a butterfly, but it's further than a specific number of meters, I'm afraid I can't remember exactly, you're not allowed to record them. And then you have to go back to the same transect several times throughout the year. And this is basically the same methodology they've used since 1976. Obviously, like, the number of transects have grown and then, you know, change around a little bit where they are, but frankly, amazing data set as well. I like data.
Yes, I think we can all stand in gratitude for everyone who has given up their time to do the UK BMS. Many research projects and research has come off the back of it, so we're all very grateful for the people that do volunteer. I think it's amazing. It's just maybe more of a step up of commitment than the iRecords, because you are sort of committing yourself to come out at certain times of the year, as you said. Okay, cool. So we've got a couple of questions in the Q&A chat. I'm just going to jump straight in, if that's okay. Our first question from Roger. Will you please define occupancy again? Thank you.
Yeah, I'm sorry, I kind of skimmed through that quite quickly. So in my case, occupancy is the proportion of all 10 by 10 kilometer squares on the island of Great Britain. If occupancy is 1 for a species in a given year, the model estimates that butterfly occurs in every single 10 by 10 kilometer quadrat square on Great Britain in that specific year. So the maximum is 1, but you could do this, and now I'm talking from my example where I'm looking at Great Britain, but if we think of it as an island, occupancy would be when a certain species is present at a certain time in all quadrats, whatever scale you use in your analyses at a given time. And if it's half, it could be, if the occupancy is 0.5, it says that's present in half of the quadrats, but it could be that it's present in the far south and the far north. Occupancy actually doesn't tell us much, doesn't give us that information. So we can have those like disjointed populations. It's kind of equivalent to range, I guess you can think of it as range, but measured on a scale from 0 to 1.
Yeah, perfect. Thanks for clarifying. So our next question is from Adam. Have you attempted any correlation between occupancy changes and habitat changes alongside climate change?
Yeah, when I first started my PhD, that would be that was the plan to be my last step. I would love to, and there's so much, as I said, there's so much to explore in this data set, and the obvious next step is to include these. A big problem is actually that we don't really have historical land use data. That data is like locked up in like physical maps. There's only like a few copies of and distributed. Yeah, we don't have that data digitized actually. But we do have the climate data, so that's the obvious next step in case I have time before I have to submit my thesis. Otherwise, I encourage everyone else to do it.
Well, brilliant. So if anyone's feeling inspired, this is clearly an area that needs to be more explored. Or
if anyone has a postdoc and want me to do it.
Yes. Galina is your girl. Cool. And we've got another question. What are some of the best ways to attract more butterflies into my garden? Any tips, flowers, etc.? Thanks for the talk. I will start tracking the butterflies I see. So we've inspired a person. I love that.
I must admit that I come from more of like a quantitative ecology background. So I really like my stats and my data. However, I would imagine that a lot of it is out of the person who asked the questions control. So if you live in a really busy city where there's no good like host plants for most species around, that's out of your control. However, if you plant different what's it called, flowers, I guess, that provide nectar for them throughout the season, I think that is an important thing. I think there's, especially around July, there tends to not be that many much food for a lot of bugs out there. Also, when you plant, think about buffering against, it might be a cold winter, it might be a warm winter. So buffer yourself again to have food for them throughout the whole flying period.
Yeah, perfect. I've also, How Does Garden for Butterflies, got a couple of links with some tips for this question E. I've just reposted them on the chat, so hopefully you can maybe have a little explore. But yeah, as Galina said, the providing nectar sources throughout the year is going to be a big factor for butterflies. But additionally, assuming you may not also have a garden, there are other ways to get involved, such as perhaps community gardens, as well as getting involved with local campaigns. So maybe to protect your local roadside verges is another really important thing. And there's some nationwide schemes as well at the moment, such as No Mow May, for people that do have access to gardens, to let wildflowers grow a bit more before we all cut them off, because we do love to mow our lawns in this country, but unfortunately it does have some devastating effects for the animals that need exactly those plants and wildflowers to eat. To survive. So yes, can get involved with that. That's a plant life scheme. So that will be on the Plant Life website if you'd like to know more about that one. Cool. So I think we all covered that. Our last question is from Sam. So Sam says, it's been really interesting hearing in this talk and the previous two that other environmental factors such as deforestation, intensification of agriculture, etc., have had potentially as much or more of an impact on butterfly populations as climate change. Do you find that it is harder to gain public interest or funding for research that is about historic changes in agriculture rather than climate change? It's a really interesting question.
It's a really interesting question. So to just address the first bit, I think whether land use change has had more or as much of an effect as climate change, I think most evidence points towards that all of these anthropogenic pressures combined risk causing a kind of like tipping point where we see where the species just can't hack all of different pressures combined at once. So it's very difficult to say, oh, one has more of an effect than the other. But to answer the last part of the question, I luckily have not had to deal with the funding, what's it called? Funding applications thus far, but I do feel like there's a lot more interest in this kind of research. So I would imagine that it's easier than it has been. And by like looking at long-term changes, that also, as I mentioned, gives us the tools to be able to better understand the changes that are going on today and make better predictions for the future.
Yeah.
Did that answer the question?
Oh yes, I think you've done a good job there. That is a very interesting question. I think you can write papers on that yourself. that could be answered in a whole essay, that one. So that is yeah, no, that we're really getting to the crux of things there, Sam. So thank you for asking that. I think that is our last question. Does anyone else have any last things? Otherwise we can wrap up, because really enjoyed hearing your talk, Galina. Thank you once again for sharing all your knowledge and wisdom and your whole process towards the digitizing the butterfly collections. It's really been interesting to hear about. I've really enjoyed that, so thank you. No problem. So this is our third talk in the Researchers' Story series. Next week, we'll have a talk from Professor Jane Hill from the University of York. And she's going to be talking to us about so following on from this question a little bit, actually, what butterflies can teach us about climate change. So if you're interested in that, tune in for next week and we can hear more about yes, exactly what we can learn about climate change and these sorts of impacts and future changes from butterfly collection. So we're not done with butterflies yet. We've got plenty more to come. So tune in for that. That's going to be next week on the 25th at 7 p.m. again. I think that is everything. There's just the questionnaire to fill out if you wish to do so. Otherwise, that's it from us. Do come along next week. Thank you all so much for coming.